Welcome to KCS at Home Summer Biology Activity 1. My name is Lisa Jacobs, and I'm a biology teacher at Hardin Valley Academy. The activities and handouts can be found under the Student Resources tab on the Knox County Schools website. We're really excited about this program, so we hope that you show us what you learn by tweeting at KCS Science. If this video is hard to understand, turn on closed captions if available, adjust the playback speed to slow down the video, consider watching short clips, then pause, listen, and watch again. Ask someone in your home to watch with you. Stop frequently and talk to your partner about what you hear and understand. In this lesson, we will sometimes pause this video to complete questions, watch another video, or follow a link. Look for the image below to know when to pause the video. You will need the KCS at Home student resources and colored pencils in blue, red, and green. So what do we need to know? The questions we're working on answering today include: how can proteins affect the way organisms look and function? Or how can they affect the phenotype of the organism? Why are some changes in our DNA passed to our offspring while others are not? And how are molecular biological techniques used, and what effects do they have on our society? You've taken a lot of biology this semester, and we're going to take a moment to remember what we've learned earlier in the year. Pause this video and follow the Quizlet link on the KCS at Home Resources #1 to practice the vocabulary before you begin. There are other review resources there as well. Welcome back. So, how can organisms look so different? These organisms are called rock pocket mice, and they live in the desert of New Mexico. As you can see, some of the rock pocket mice live in sandy areas with light-colored sand, while other rock pocket mice live in areas where dark rocks were formed by lava flows thousands of years ago. Take a moment and pause this video. Use the link in #2 to watch Natural Selection and the Rock Pocket Mouse. Then share your ideas by answering question #2. Why do you think most of the rock pocket mice living on sand look different than most of the rock pocket mice living on the dark rocks formed by lava flows? Welcome back. DNA is a nucleic acid. And let's review the role of DNA. One of the roles of DNA is to store genetic information. For example, a flower may have genes, which are segments of DNA, for purple pigment. Another role of DNA is to copy itself. Every time a cell divides, it must make an exact copy of the purple pigment gene. And finally, the role of DNA is to put genetic information to work. Cells use instructions from DNA to produce proteins. In our example, the cells of the flower would use the instructions in DNA to produce enzymes that make the purple pigment. Information in DNA is stored in the sequence of the nitrogen bases. You can see the nitrogen bases here in the center of the DNA molecule. The base adenine, or A, always pairs with the base thymine, or T, while the base cytosine always pairs with the base guanine, or G. So those are some cumbersome words. We usually just use their letters to refer to them. And we can remember them by thinking about A and T. Apples grow on trees. And C pairs with G, like cars park in the garage. This brings us back to our first question. How can proteins affect the way organisms look and function? In molecular biology, the central dogma describes the flow of information. Information flows from DNA to RNA to protein. Another way of saying this is to describe protein synthesis. During protein synthesis, DNA undergoes a process called transcription to make mRNA, which undergoes a process called translation to make a protein. When a gene, or a small segment of DNA, is used to build a strand of mRNA, which is used to build a protein, we say that that gene has been expressed. So why is protein important? Remember back to the beginning of the year when you studied biomolecules like carbohydrates, lipids, proteins, and nucleic acids. Hopefully you remember that proteins are responsible for almost everything in an organism. They have so many functions. They regulate cell processes, they control reaction rates, they transport substances, and they help fight disease. They also form structures like the fur on a puppy, or the muscles that allow you to move, the cytoskeleton inside of your cells, your fingernails and toenails, and even the color of the fur on a mouse. Pause this video and take a moment to answer question number 6. RNA is also a nucleic acid like DNA. There are several kinds of RNA, but we're going to focus on messenger RNA, or mRNA. Pause the video and study the diagram in number 7. List the differences and similarities that you see between DNA and RNA in the table provided. Let's check your answers. DNA and RNA have some similarities. They both have sugar phosphate backbones, and they both contain the nitrogen bases guanine, cytosine, and adenine, C, G, and A. But they also have some differences. DNA is double-stranded, while RNA is single-stranded. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. But the most important difference for our purposes when talking about protein synthesis is that in DNA, the base adenine pairs with the base thymine, A to T. Whereas when we're building RNA, the base adenine pairs with the base uracil, or U. Let's take a moment and focus in on RNA, or messenger RNA. Messenger RNA is a copy of one section of DNA, or a gene, that can leave the nucleus and be used to make a protein. This genetic code is read in 3-letter groupings called codons. For example, the first codon here is AUG, the next codon is AAC, and the final codon is UCU. Each codon in messenger RNA codes for one specific amino acid. We can use a codon chart to decode a sequence of mRNA and find the amino acids of the protein that will be produced. Let's look at our example. Start at the middle of the circle with the first letter of the codon and work your way out. For example, if we'd like to find the amino acid that is specified by AUG, I would start in the center to find the A. Moving out, I would find the U and the G. And this would lead me to the amino acid methionine that matches with AUG. Take a moment and pause your video. Use your codon chart to find the amino acids specified by the codons AAC and UCU to complete number 8 in your KCS at Home resource. Finding the amino acid specified by AAC, we would start in the middle, find A, move out to the next A, to C, and that would lead us to the amino acid that is specified by AAC, asparagine. To find the amino acid specified by UCU, we will start in the middle at U, move out to C, out to U, and in our final circle, we find the amino acid serine that is specified by UCU. While we continue to answer our first question, we are also going to find out how to answer our second question. Why are some changes in our DNA passed to our offspring while others are not? Let's go back to the desert to find out. Pause this video and use the link in number 9 to watch the rest of Natural Selection and the Rock Pocket Mouse. After you finish the video, read Molecular Genetics of Color Mutations in Rock Pocket Mice. Welcome back. Now we're going to practice transcription, the process of copying a base sequence from DNA to RNA. In the video, you saw that the scientists did a lot of work to find the sequence of the genes that caused the color differences in rock pocket mice. What they came up with were some mutations in the gene called MC1R. We're going to work in number 10, so follow along in your resource to practice. The wild-type MC1R gene results in the light-colored coat phenotype, while the mutant MC1R gene results in the dark-colored coat phenotype. The scientists in the video used biological technologies such as polymerase chain reaction, or PCR, to make millions of copies of the rock pocket mice DNA, as well as gel electrophoresis to analyze the sequence of bases. What we have here is a small segment of the MC1R gene, which has close to 1,000 bases. This small segment shows codon number 15 through codon number 24. Looking at the DNA sequence of the wild-type gene and comparing it to the DNA sequence in the mutant gene, you can see that there is one mutation in this segment. In transcription, we'll use the DNA sequence to code for the mRNA sequence. The first DNA bases are T, T, and G. We know that T always pairs with A, so we have A, C. This is the tricky part about transcribing to mRNA. Whenever you see an A in DNA, it means you'll have a U in RNA. So our second codon will be UCC. We can fill in the rest of the codons, ACC, CGC, ACA, GGC, GUU, CCU, CAC, and CUC. When we work on transcribing the mRNA in our mutant MC1R gene, recall that there's only one mutation in our DNA sequence, so all of the other codons will be the same. So we can go ahead and fill those in from the wild-type gene that we've already transcribed. And we just need to transcribe the ACG into UGC. Now we're moving on to the process of translation, the decoding of an mRNA message into a protein. Follow along with number 10 in your resource to practice. We're going to look at the wild-type MC1R gene and use the mRNA sequence to find the amino acid sequence. You'll need to go back to your codon wheel and find AAC, which will result in the amino acid asparagine. Use your codon wheel to fill in the rest of the amino acid sequence. When we look at our mutant MC1R gene, remember that only one of the codons in our mRNA is different. So when we find our amino acid sequence, we will only have one amino acid that could be changed. So we can go ahead and fill in all of the rest of our amino acids from the work we did above. Then go back to your codon wheel and look for UGC, which results in the amino acid cysteine. Comparing it to the amino acid sequence above, you can see that the amino acid has changed. We've created a missense mutation. Now it's time for you to practice. Pause this video and find the mRNA sequence for both the wild type and the mutant gene MC1R segment in number 11. Let's check your answers. Here's the mutation, the difference between the wild type and the mutant type. Filling in your mRNA sequence gives you codons GUG, CAC, AUG, CUU, GCA. Filling in the mRNA sequence for the mutant gene, remember that the only codon that will be different is the one where we have the mutation in the DNA. So we can fill in all of the rest of the codons, just copying them from above, and find that the new codon in our mutant type is CUC. Now let's practice translation. Pause this video and find the amino acid sequence for both the wild type and the mutant MC1R gene segment in number 11. Let's check your answers. The amino acid sequence is valine, histidine, methionine, serine, and alanine. Remember, when we're finding the amino acid sequence in our mutant gene, we will only have one difference where we have the mutation. So we can fill in all of the rest of the amino acids based on the work we did above. Going back to our codon wheel, we can find that CUC specifies serine. Notice that the amino acid above is also serine. So although we had a mutation in the DNA, it's resulted in the same amino acid sequence. We call this a silent mutation. Pause the video again and complete the mRNA sequence and the amino acid sequence for both the wild type and the mutant MC1R gene segment in number 12. After completing the transcription and translation of the MC1R gene segments, read types of mutations. What is a mutation? Mutations are any change in the DNA sequence. Are they helpful? Are they harmful? Are they neutral? Mutations can be any of those things depending on what the result is and the environment that the organism lives in. Often when people hear the word mutation, they think of things like aliens and crosses between animals, this cute guy, and even these pizza lovers. At the top, you can see a normal sequence of DNA before any mutations have occurred. Once that DNA has undergone transcription and translation, the amino acid sequence is shown above. There are three types of mutations that we're going to talk about today. The first is a substitution, where one base is substituted for another. In the original, there was a C, or cytosine, but after the mutation, there is a T. And you can see that that has resulted in a different amino acid sequence. Next, we have an insertion, where an extra nucleotide is put into the DNA sequence. This shifts the frame of reading, and so all of the codons after that insertion are also different. You can see that this results in many different amino acids after the mutation. Deletion also results in a frame shift mutation. When a nucleotide is removed from the sequence, that causes the reading to shift again and can result in different amino acids after the mutation, as well as the possibility of an early stop signal that makes the protein too short. These types of mutations can result in a silent mutation, as we saw earlier, where there's no change in the resulting protein, a missense mutation, where the amino acid sequence is changed, resulting in a change in the structure of the protein. And when the structure of a protein changes, remember that that changes the function of that protein as well. Or a nonsense mutation, causing an early stop signal that results in a shortened protein. What do you think? Which of these mutation types do you think is most likely to be harmful? Pause the video and complete questions 13 through 25. You can refer back to the Rock Pocket Mice video as needed. This brings us to our final question. How are molecular biological techniques used, and what effects do they have on society? The field of genetic engineering has experienced enormous advancements in recent history. In our discussion of the Rock Pocket Mice, we saw that the scientists used polymerase chain reaction, or PCR, to make millions of copies of the DNA segments needed to study the genes that resulted in the different fur colors. They then took those DNA segments and analyzed those DNA segments with a process called gel electrophoresis. Gel electrophoresis allows scientists to find out which sequences of nucleotides are present in a DNA strand. Humans have also used recombinant DNA techniques for quite a few years. An example of this is when humans took a portion of a gene and implanted it into a bacterial cell so that that bacterial cell could produce human insulin. Insulin is a hormone that is used in the human body and is also needed to treat people with diabetes. We also have created genetically modified organisms. Many of the crops that we eat on a daily basis have been genetically modified, oftentimes to make them resistant to particular types of insects. And finally, that brings us to the newest technique called CRISPR. CRISPR is a tool that can directly change the DNA base sequence of a gene. It is much faster and cheaper than any of the techniques that have come before it. And it is also much more accurate. It has opened up an entirely new set of ethical questions. We have the ability at this point to cut out a very specific portion of a gene or to change a very specific portion of a gene. So the question becomes, just because we can, does that mean we should? After you have watched this video, read the article, The Many Efforts to Lick Cat Allergies, and answer the rest of the questions in your KCS at Home resource. This week, we answered the questions, how can protein affect the way organisms look and function, by looking at the rock pocket mice living in New Mexico with their different fur colors. Why are some changes in our DNA passed to our offspring while others are not? Remember that mutations in a somatic cell or a body cell cannot be passed on to offspring. Mutations that will be passed on to offspring must occur in a germline cell or a sperm or egg. And how are molecular biological techniques used, and what effect do they have on society? I hope that you look further into the technique of CRISPR, as it is something that will be around for a long time and could have huge impacts on our world. Next week, you will answer the questions, how is all life the same? When you revisit the rock pocket mice, you will find out what has led to differences among living things, how do populations change over time, How do new traits appear and become more common in a population? Thank you for joining me, and remember to show us what you learned by tweeting at KCS Science. Have a great summer.